population that was 25 homozygous dominants, 18 heterozygous, and 3 homozygous recessives. Who has a calculator can grind out those genotype frequencies to me? Not the allele frequencies, but the genotype frequencies. Well, what's our grand total now? 46 individuals? Does that sound right? So if I say, what is the genotype frequency of homozygous dominant? You'd say, well, there's 25 of those individuals who are big Q out of 46 individuals. And so that comes out to, did anybody do the math yet? Yeah, 0.54 is the genotype frequency for homozygous dominant. And for heterozygous, 18 of the 46 individuals are heterozygous. It's what? 0.39. Yep, that sounds good to me. And the homozygous recessive individuals, 3 out of 46 are homozygous recessive. And that turns out to be 0.06, okay. 3 divided by 46. Yes, 0.06. Okay, so hopefully you'll be able to handle it when I give you examples that don't neatly add up to 100%, to 100 individuals. You just find their percentage out of whatever the total number of individuals are. But let's go back to this easier example where it did come out to 100 individuals. All right, so I think one of the reasons why students get confused in this population genetics lecture is they mix up the allele frequencies with the genotype frequencies. So let's take a moment to appreciate what these are. The genotype frequencies just say what portion of the entire population, the 100 individuals, has a particular genotype. And there's only 3 genotypes: big B, big B, big B, little b, little b, little b, homozygous dominant, heterozygous, homozygous recessive. That part's, I think, fairly straightforward. Don't confuse that with allele frequencies, where you talk about the number of alleles, which is always twice the number of individuals, and you have to sort of extract out of the numbers here how many big B alleles there are, how many little b alleles that there are, remembering that these guys actually there's 12 big Bs from this group and 38 big Bs from this group, so you get a total of 50 big Bs, and there's 38 little Bs from this group, but 112 little Bs from this group, because they have 2 little Bs, so you end up with 150 little Bs there. It's a little more complicated to do the allele frequencies, but the point I'm making here is don't mix up questions that ask you for allele frequencies with questions that ask you for genotype frequencies. All right, so far? Okay, now, let's get back to the elephant seals again for a moment. Let's see, there we go. No, there we go. When the laws of genetics were first being discovered, or I should say after they were discovered and people began to talk about them and think about what they meant, people sometimes said, why don't the dominant alleles eventually make the recessive alleles disappear? Because, I don't know, if you think about wild animals for a second, the stronger ones dominate the weaker ones and maybe eventually drive them out of the population or kill them off. And so people said, why don't the stronger dominant alleles kill off the weaker alleles in the population? And it turns out that doesn't happen. And I'll give you an illustration here. Let's just say, for the sake of argument, that we're going to mate these 2 plants with each other. One of them's purple, big P, and one of them's white, little p, little p. If our entire population is these two plants, what is the allele frequency of the big P and what's the allele frequency of the little p? So we have two individuals. How many alleles do we have total? 4, right. You always double the number of individuals tells you the number of alleles. So we have 4 alleles present here total. Of those 4 alleles, how many are big P? 2. And how many are little p? 2. So 2 out of 4 and 2 out of 4 is 0.5. And so our allele frequency, maybe I'll write it over here, for big P, allele frequency is 0.5, and for little p, allele frequency is 0.5, like so. Okay, now we're going to mate these plants with each other, and we've done enough Mendelian genetics. That's pretty straightforward. We make our Punnett square, we draw the gametes there from that plant and the gametes there from that plant, and then we run them together, and this tells us what the offspring are going to be. And let's say that they had a total of 4 offspring. Now what are the allele frequencies? Nothing's changed. We have now 4, let's just focus in on the F1 generation inside the Punnett square. We now have 4 individuals in the population. They're all big P, little p. And if there's 4 individuals, there's 8 total alleles. But 4 of those alleles are big P and 4 of those alleles are little p. So we're right back to where we started. In the F1 generation, the frequency of the big P is still 0.5, and the frequency of the little p is still 0.5. In other words, half are big P, half are little p, which is the same as what we had in our starting generation, in the parental generation. All right, now we're going to take the F1 generation and mate them with each other. And there's the Punnett square for that. So now let's look at the F2 generation that we produced. I'm not there. Here it is. Here's the F2 generation. We mate the F1s with each other. There's the Punnett square. So let's say the 4 Punnett square children are our new population. Now what are the allele frequencies? Nothing's changed, yeah. If you look at the offspring, and if this is now the new population, we have 4 individuals, 1, 2, 3, 4, so 8 alleles total. How many big P's do we have? 1, 2, 3, 4. How many little P's do we have? 1, 2, 3, 4. Nothing's changed. And so that's what they discovered. Despite the fact that these are dominant alleles and these are recessive alleles, nothing changes. They're being reshuffled in different ways. You know, the initial population was half homozygous dominant and half homozygous recessive, and then the F1 generation was all big P, little p's heterozygous, and the F2 generation is now 1 part big P to 2 parts big P, little p to 1 part little p, little p. The genotype frequencies are changing around. You know, how many are big P, how many are big P, little p, how many are little p, little p. The genotype frequencies are changing around, but the allele frequencies aren't changing a bit. And so that was a major conceptual breakthrough, that as long as mating is random, in other words, as long as the purple plants aren't purposefully mating only with purple plants and white plants with white plants, or as long as the black-eyed elephant seals are not choosing only black-eyed ones to mate with and not brown-eyed and things like that. If the mating is completely random and the alleles are equally beneficial, as long as you're not harmed by being brown-eyed or black-eyed or purple or white. If everything's equally beneficial to you, then the allele frequencies just don't change. The genotype frequencies might, but the allele frequencies don't change at all. Does that sound reasonable? Maybe a little interesting, but hopefully from the plant example we just did on the board here, you can say, oh yeah, I guess that's true. Okay, so we have these 2 different concepts here, the allele frequencies and the genotype frequencies. And as I just showed you with that Mendelian cross, we saw the genotype frequencies changing from the parental generation to the F1 to the F2, but the allele frequencies didn't change. But something interesting would happen. If we kept mating those plants with each other, we went to the F3 generation, the F4 generation, the F5 generation. If we kept meeting them with each other, yeah, the allele frequencies would never change. They would always stay at 0.5, as long as the mating was random and the gene being purple or white didn't change your survival. We would always be at 0.5, 0.5, 0.5. But what's interesting would happen is eventually the genotype frequencies would settle down. Remember, I showed you that the genotype frequencies were changing from one generation to another, but eventually they would settle down to a certain percentages and stay at those percentages. And that's called coming to equilibrium, or sometimes coming to Hardy-Weinberg equilibrium, after the people who first discovered this. And so what I'm going to do now is show you what that equilibrium is. In other words, if these elephant seals kept mating with each other, what would their genotype frequencies settle down into? As a matter of fact, I think I already gave you the answer right here. Here's the equation that you use. In other words, this is the equation that relates the allele frequencies to what they eventually settle down into in terms of the genotype frequencies. And it looks like this. If you take the frequency of the dominant allele and square it. Oh, actually, before I do that, let me go back over here. These have to equal to 1 when you add them all up. Because these are the only 3 possibilities, and all the possibilities eventually have to add up to 1. Anyway, if you take the dominant allele frequency and square it, that gives you the homozygous dominant frequency. If you take 2 times the dominant allele frequency and the recessive allele frequency, that gives you the heterozygous genotype frequency. And if you take the recessive allele frequency, that we calculated over there, and square it, that gives you the homozygous recessive genotype frequency. And let's verify that for a second. If we were saying that the frequency of the dominant allele is 0.25, if you square 0.25, I bet you come out with 0.06, or at least something close to it. Yeah, within the margin of error of the calculator, that did give me the frequency of the dominant allele, or the homozygous dominant genotype. And if we take 2 times the frequency of the dominant allele and times the recessive allele, that should give us that 0.38 of the heterozygous genotype. So I take 0.25, well, I should start off with take 2 times 0.25, the dominant allele frequency, times 0.75. Yeah, it's, well, 0.375, which is rounding close to 0.38. And for the homozygous recessive, that's the 0.75 squared, and it works out. It comes out to 0.56. And this is yet another equation that adds up to 1. If you, well, I guess that's the same thing here. That the frequencies, the genotype frequencies must add up to 1. Put the equals 1 sign. And so this equation that I put in the center of the board, think of that as now you're able to link the allele frequencies, the big B and little b frequencies, to the genotype frequencies. What percentage of the population is homozygous dominant, what percentage is heterozygous, what percentage is homozygous recessive. As long as they're mating randomly and the alleles don't cause any survival benefit versus the other one, if the alleles are equally survivable, so to speak, the genotype frequencies will eventually settle down into the ones predicted by this equation. And it's called the Hardy-Weinberg equilibrium equation, after the two mathematician biologists who discovered it. And the only thing confusing is if you look at it in a textbook, instead of saying the frequency of the dominant allele squared and the frequency of the recessive allele squared and 2 times dominant frequency and the recessive allele frequency, they use the letters P's and Q's, where P is the allele frequency of the dominant allele, big B, F, and Q is the frequency of the recessive allele, little bf. But I think that's kind of confusing because it makes them almost look like, well, I don't know what it makes them look like, but I find that confusing. And so if you want to think about it with the big B's and the F and the little bf's, that might make more sense to you than it does to me. All right, so let me see if you can follow me on this. I'm going to give you a different example here. Let's say that we're studying some population, and bust out your calculators and see if you can get this. And in this population, we find 162